Hello guys, how are you? I hope you are doing fine. Today, the topic of my presentation is metabotropic glutamate receptors. Metabotropic glutamate receptors are very important in brain for the excitatory neurotransmission. So mostly we all know about the ionotropic glutamate receptors, but this presentation is specially dedicated for metabotropic glutamate receptors, their role and their significance. So the lecture contents are location of metabotropic glutamate receptors. They are located at the glutamatergic synapse. Difference between ionotropic and metabotropic glutamate receptors, and the location of metabotropic glutamate receptors at their synapse and their role in the neurotransmission. That these are three major points that I am going to cover in this presentation. So let's first of all talk about a glutamatergic synapse. The glutamatergic synapse is basically shown here, which is composed of a presynaptic terminal and a postsynaptic cell. As we all know that the synapses, whether they are excitatory or inhibitory, they are made by a presynaptic terminal, a coming from a neuron from the presynaptic terminal, and they make connection on another neuron, and this like postsynaptic is known as postsynaptic component, and the receptors, they are expressed on the postsynaptic area. For example, in this case, the glutamate receptors, they are shown here. So basic scheme of glutamatergic synapse looks like this, where the excitatory amino acid transporters, they take up glutamine, and with the help of this enzyme glutaminase, it is converted into the glutamate, and then it is packed into the vesicles for the release at the time of release, when glutamate will be needed to be released by the synaptic release neurotransmitter release mechanism at the synapses. So this is VGLUT, basically stands for the vesicular glutamate transporter, which basically transports the glutamate into the vesicles and keeps them ready for the release at the time of need. So once glutamate is released, it acts on the postsynaptic receptors. So there are different types of receptors, glutamate receptors. Here, if we talk about, they are shown the ionotropic glutamate receptors. They are shown here. So the termination of the glutamase is also necessary. What are the pathways of termination of glutamate action? So the important one is EATT, excitatory amino acid transporter. So it takes up glutamate into the glial cells. This is very important function performed by the glial cells shown here, and then it is again glutamate is converted into the glutamine and then comes back to the presynaptic terminal. So this cycle goes on. So this is very brief about the glutamatergic synapse physiology. Okay, now coming back to the glutamate receptors again, like we have discussed, so there are two types of glutamate receptors: ionotropic glutamate receptors and metabotropic glutamate receptors. The glutamate and GABA are the major neurotransmitters in the vertebrate central nervous system. The glutamatergic receptors can be divided into two broad categories, like the ionotropic receptors and metabotropic receptors. Ionotropic receptors are further subdivided into three types: AMPA receptors, kinase receptors, and NMDA receptors. I will make a separate lecture on the ionotropic receptors because they are also very important. So what is the difference between ionotropic glutamate receptors and metabotropic glutamate receptors? That you can see here on the right side in the diagram. Ionotropic receptors are those receptors when glutamate binds at the binding site, it results in opening of an ion channel and results in depolarization. So the example ionotropic receptors are non-NMDA receptors and NMDA receptors. As I have told you, I will make separate lecture on ionotropic receptors. So today's focus is on the metabotropic receptors. Metabotropic receptors are the type of glutamate receptors where when the glutamate as an agonist binds, and then it results in activation of G protein. So basically, the metabotropic receptors, they are basically G protein coupled receptors. As you are know that the G protein coupled receptors is one of the famous types of glutamate, is one of the famous types of the family of the receptors. So once these receptors they are activated, it results in the activation of G protein, and then G protein actually then activates PSA phospholipase C, and then it results in the formation of inositol triphosphate, which is shown here. Inositol triphosphate and DAG signaling pathways stimulated. So the important point is the action of glutamate on the ionotropic receptor is always excitatory, while the activation of metabotropic glutamate receptors can produce either excitation or inhibition. So this is something very interesting fact about the metabotropic receptors, that they can be inhibitory as well. So I will show further details about these. So further, if we talk about the classification of glutamate receptors, the ionotropic receptors, they are shown here, and the metabotropic receptors, they are shown on the right side. So this classification is arranged based on the group name and the subunits composed of and the physiological function. And you can see that the ionotropic receptors, they can be NMDA, AMPA, and kainate, and their units, subunits, composing subunits, they are shown here. And then metabotropic receptors, they are divided, they can be given the name of group 1, group 2, and they are divided into three groups, group 1, 2, and 3. And further, if we talk about the subtypes of the mGluRs, they are like 8 subtypes. Group 1 is the difference between these three groups is that the group 1 mGluRs, they are slow excitatory, and the group 2 and group 3 metabotropic receptors, they are slow inhibitory. On the other hand, ionotropic receptors, they are all fast excitatory. So further looking at uh, the metabotropic glutamate receptors subtypes, group subtypes, the group 1 results in activation of IP3 and DHE pathway, like I have explained just a while ago. So they are also, like I have also explained, that these are G protein coupled receptors. So this is the agonist binding site, and this is the intracellular site where the activation of G protein results in the activation of those second messenger cell signaling pathways. Similarly, activation of group 2 receptors results in decrease in cyclic EMP, and similarly, the activation of group 3 receptors results in decrease in cyclic EMP level. So that is how they are responsible for the slow inhibitory response. Now, further, if we talk about the location of metabotropic glutamate receptors, we will understand that the how their second messengers can be excitatory, can help them make excitatory or inhibitory. For example, a glutamatergic synapse is shown here. This is the presynaptic component, and this is the postsynaptic component. Postsynaptic neuron or postsynaptic terminal is shown here. So you can see that the glutamate group 1 glutamate receptors, group 1, like mGluR1 and 5, GluR1 and 5, they are mostly located on the postsynaptic spines. So you can see that the location of mGluR1 type group 1, they are located on the postsynaptic receptors, and activation of these receptors would result in regulate the postsynaptic ionotropic glutamate receptors as well as potassium and calcium channels. So once glutamate binds over here, it actually acts on the calcium channels as well as on the AMPA and NMDA receptors. So binding of glutamate on these receptors would result in excitatory response. On the other hand, mGluR 2, group 2 and group 3, they are located on the presynaptic terminus. So this presynaptic terminal, once the glutamate comes and binds on these receptors, stimulate these receptors, presynaptic inhibition takes place, and it results in the inhibition of glutamate release, and as well as it acts on the like these receptors, they are shown here, they act on the calcium channels. So uptake of calcium into the presynaptic component is decreased, so they show an inhibitory response. So group 2 and group 3 receptors, especially for group 3 receptors subtypes 4 and GluR4, 7, 8, are mostly presynaptic and regulate neurotransmitter release at least by inhibiting calcium channels. Okay guys, thank you. So this is all for today. Thank you so much.